Prostate cancer is the leading cause of cancer incidence in men. Despite increased research into treatment options in recent years and much greater prognosis, prostate cancer remains the second highest cause of cancer-related deaths in men worldwide. As such, it is imperative that therapeutic agents that fight prostate cancer are continually proposed, trialed, and developed. There are many pathways which can lead to the development of prostate cancer. This video will focus on the development of tumours through androgen receptor activity and signaling. Androgen receptor signaling is important for the proliferation and survival of prostate cancer cells. The majority of castrate-resistant prostate cancer cells have been shown to still maintain androgen receptor-mediated signaling activity and dependence. The level of prostate-specific antigen, a product of androgen receptor signaling, can be seen to rise during the progression phase of each androgen suppression cycle. This demonstrates the dependence of prostate cancer cells on the androgen receptor. On top of sustained proliferative signaling, mutant androgen receptors can also occupy the promoter region of the human telomerase reverse transcriptase, or hTERT, and increase its transcription. This can be seen from the fact that the mRNA level of hTERT significantly decreases when androgen receptors in LN cap cells are knocked out by the addition of MSA and doxysilane. hTERT is a catalytic subunit of active telomerase, and its transcription can enable prostate cancer cells to overcome replicative senescence, leading to their immortality. Despite the many pathways leading to prostate cancer development, we believe we have a novel therapeutic agent that will prevent androgen receptor signaling-induced prostate cancer development. MicroRNA-mediated post-transcriptional regulation has been discovered to be essential for many biological processes in the body, and in this video we will show you how this regulation can play a role in treating prostate cancer. We propose using MIR145, a type of microRNA, as our main therapeutic target. When this microRNA is delivered to the prostate cancer cells, it can bind to AGO2, a type of protein required for gene silencing. This will activate the RNA-induced gene silencing complex. This complex then moves towards the androgen receptor transcript and binds to it, resulting in cleavage. This shows how MIR145 can reduce expression of AR and its downstream signaling pathways through gene signaling and help to reduce the proliferation of androgen-dependent prostate cancer cells. On top of this, downregulation of androgen receptor expression would in turn reduce its occupancy at the hTERT promoter region. This would decrease hTERT transcription, which would therefore prevent telomerase from constantly extending the chromosome of prostate cancer cells, thus preventing immortality. MIR145 will be targeted to prostate cancer cells through lipofection. Lipofection is a transfection technique where lipid RNA vesicles are formed through interaction of liposomes and RNA. The liposome for delivery of MIR145 will be made of folate conjugated lipids and will be administered by intravenous injection. As folate receptors are overexpressed on cancer cells, the liposome is targeted directly to cancer cells through the folate conjugated lipids that make up the liposome. Upon binding to folate receptors, the liposome fuses with the cell membrane, and the exogenous MIR145 is taken up by the cancer cell through endocytosis, and is subsequently taken up by the prostate cancer cell nucleus, where it binds and blocks androgen receptors. Through blocking androgen receptors, the exogenous MIR145 enclosed liposome attacks prostate cancer on two separate hallmarks of cancer, sustaining proliferative signaling and enabling replicative immortality. We believe this novel dual-pronged treatment of prostate cancer will be more effective than other drugs currently on the market. Whilst we have a viable idea for the successful treatment of prostate cancer, further research must be conducted on how to avoid blocking androgen receptors on non-cancerous cells that are androgen dependent. It is estimated that $2 million is required to put the exogenous MIR145 enclosed liposome through further research and clinical trials, with a possibility of being available to the public by the year 2020. With your support and funding, our prostate cancer therapeutic agent can be put into reality.